Yeah. So can you see my screen?
Yes, ma'am.
Okay. So in the last session we have saw like about the mapping, right? We mapped our reads on the index genome. So in the mapping, what we saw, we were adding some options, that is the DTA cufflinks. So before we today we install the cufflinks tools, I want to tell you about the DTA cufflinks here. So this DTA cufflink stands for tailored alignment for cufflinks. So why this option is designed, actually it is just to optimize this HISA2 tool, or you can say the HISA2 alignment output, which is specifically for compatibility with the cufflinks transcript. So by using this DTA cufflink option, you can enhance the accuracy as well as the efficiency of the cufflink transcript assembly process. So which is leading to the more reliable gene expression as well as their analysis result. So when this DTA cufflink is enabled, so basically it is focusing on the region that are more likely to the transcript boundaries and the alternate splicing events. And one thing more, it is improving the cufflinks performance, which is basically minimizing the false positive results, or you can see the alignment. And this option is beneficial for identifying the alternative splicing events, or you can see the transcripts. Is it clear what is this TTA cufflinks now? Why it is designed in this tool?
Yes, ma'am. Got it.
So now we need to install this tool, the cufflinks. Just open this cufflinks folder. Open. You need to respond.
Yes, ma'am.
So then from here, just open the terminal.
Done, ma'am.
Done, ma'am.
Then just write here pwd, because we need to copy the path of this cufflinks folder. Okay. So you can see this is the path here. From here, you need to copy this and paste in your script. Okay. And just write the path of the link here. Make sure the path should be there, like where your files are present, the cufflink, cuff merge, cuff norm, this thing. Okay. Pasted.
Yes, ma'am.
Okay. Then just go to this raw data, and from this raw data you need to open the new terminal again. And there is one command you just write here, sudo, give the space, nano space slash etc bash dot bashrc bash rc file. So what is a sudo? It is a root ss here, and the nano is a text editor. Whereas this etc is the folder where is the bashrc file written.
Yeah, ma'am.
Then enter and give the password. Just open this file.
Just a minute, ma'am. Can you tell me the command again? Actually, my WSL
Yeah,
wasn't.
yeah, sure. Um, I will just send you in the text chat box. Sudo
Done, ma'am.
done. Okay. So now are you on the same page, everyone?
Yes, ma'am.
So now what you have to do, just scroll down till you find out the F1 key. Okay? Just go till the end, okay? And you will see this F1 in your file, this F1. Can you?
Yes,
Yes,
ma'am.
ma'am.
Now here you have to write export, give the space, then put the capital letters in the path equal to $ path and then colons. Just write here till the colon.
Yeah, done, ma'am.
Done. So now in the script you have pasted the link of that folder, that Cufflinks folder, correct? So that you need to copy from the script, okay, this, and paste that command in this bashrc file. Give the path.
Yeah, done, ma'am.
Okay. Everyone?
Yes, ma'am.
Yes, ma'am.
Okay. Then just type Control X, then Y, enter.
Yeah, done, ma'am.
Done. Then just cancel this and you have to open the new terminal again from this raw data. Just type here WSL and write here Cufflinks and the manual will be opened.
Yes, ma'am.
Pushpit, it is done.
Yes, ma'am.
Arya,
Yeah, ma'am, it's done.
Sonali, Juhi, Namarta, Umi.
I am facing some problem. That last if that you said, I'm not able to write after that.
You just share your screen, Sonali.
Yes, ma'am.
Can you just maximize this? Yeah, this is the F1. Go down. Can you see this F1? Yeah, just enter. Hello?
Yeah, just a minute. When I enter, it is going up first line.
Just open the file again. Just cancel it. Just write the command again.
So I just
Close this. Yeah.
close this. Okay.
Actually, in the bash RC, you don't need to do any changes. Otherwise, tool won't be able to run in your system. Okay. So be careful with this bash RC. So yeah, you open. Just write here sudo space nano space slash etc etc, yeah, slash bash dot bashrc. Enter. Do it from the keypad. Yeah, go down. Yeah, here it is. Export zip space path equal to dollar path. Uh, you just have to write Sonali here again. The command export. Yeah, just write it below this fast QC. Yeah, give the space path. So now the path of the cufflinks you have to paste here. Yeah. So now just type Control X.
I have to type or
No. Just from the keyboard you have to press that Control X. Then Y. Press Y. Then enter. Yeah. Just now just cancel this terminal. Yeah. Now again open the new terminal from here. Raw data. Now just write a cufflinks. Enter. Yeah. Now can you guys just write here cuff merge? Okay, so there is an error. I will tell you. Just others can also try. Just write here cuff merge.
I'm also getting an error for cuff merge.
Okay, I will tell you.
Ma'am, it's showing no such file or directory.
Yeah. Everyone it is getting.
For me, it's something different. It's showing except get op dot error message syntax error multiple exception types must be parenthesized.
Okay. So you need to install the Python version 2. If you just copy this error, Sonali, can you just copy that? User bin Python no such file or directory. Just paste in the browser there. Yeah, go down little bit. Actually, why I'm telling you, so whenever in the installation part you are getting error, you need to copy this error and you have to paste in the browser. Okay. So for this error, I have already figured this out. What is the problem? So in your system, guys, like the Python version is not there. Okay. Python is not installed. So you need to install the Python 2 because the cuff merge require the Python 2. So can you just install the Python 2 sudo apt hyphen get install Python 2? Why? If the error is same, so everyone should install this Python version 2. Done? Okay. Wait, Sonali. Everyone did?
Yes, ma'am.
Sana, Yash, Pushpit, Namrata, Juhi. What about you guys?
Yes, ma'am.
Yes, ma'am. I was having different errors, so
Who? Juhi.
yeah.
Juhi, we will discuss later. First, let them solve this. Okay? We will see your error. So yeah, let me share my screen now.
Has stop written.
Uh, but ma'am, now also after
Wait,
entering,
wait. Arey, I will tell you. Wait, wait. Can you see my screen?
yes, ma'am.
So now cuff merge, I have written cuff merge require which version of Python? So it is requiring the Python 2. So now if you open this tool folder and you go inside this cuff merge file, just open this, and in the user bin environment, you have to write here Python 2. Then save this file.
Ma'am, can you repeat? I didn't want it.
Yeah. See, uh, just go inside this cufflink folder. Everyone is having, right? Cufflink folder. So inside that, you can see there is a cuff merge file. Everyone can see that?
Yes, ma'am.
Yeah. Then just open this cuff merge file and you will see at topmost there is a user bin environment. So we are creating the environment of Python 2 there. Just type this Python 2 in your file. Once it is done, then save this file.
I mean, mine is written Python only, not Python 2.
Who? Sonali?
Yes.
Uh, just share your screen, Sonali.
Done, ma'am.
Done. Okay. Arya, now you just open the new terminal from inside the raw data and just type the cuff merge there.
Yeah, ma'am, it's opening. I checked.
Yeah, and just write cuff diff now. Is it opening? Yeah, ma'am. Can you see that user bin environment Python? Just type here
Yeah.
2 here. No. Type here 2 beside the Python. Right. Yeah, save this file now.
Yeah, ma'am, curve diff also is opening.
Okay, Arya, done it's for you. Yeah. Just close this. Now, just open the new terminal from this raw data. Go to raw
Ma'am,
data.
in mine, when I typed cuff merge, it is saying user bin
Yeah.
environment Python,
Pushpit,
no such thing.
wait. Just type here, ma'am, cufflink, cuff merge, sorry. Merge. Enter. Yeah. Cuff diff. Just type. Yeah. Done. So when the manual is open, so successfully installation is done. So Pushpit, you can also share your screen now. Pushpit.
Yes, ma'am. Is it visible?
Mm, yeah. So you haven't installed the Python 2 here in your system?
Okay, I also have to do it. I thought my cufflinks is working, so I don't
No,
have.
See, the thing is for the cuff merge, the requirement is the Python 2. Actually, the trainer might have taught you like in the Linux session also, like the tools are depending on the packages also. Okay, it's
Mhm.
not about like when you are downloading the zip files, it will install in your system. No, it never happen in the source code. Okay, so in the source code, you have to just read your help me file also in order to get those packages. So there are a lot of other packages also you need to install before the proper installation. Okay, so yeah, you can just try Pushpit, just download that Python 2, then open the cuff merge file, then type there Python 2, then save it. So Juhi, you can share your screen.
I followed the same steps now. It's open. It's opening.
Yeah, okay. So ma'am,
Okay.
every like file it is opening, cuff merge, cuff diff.
Yes, all
Okay.
files are open.
Okay, fine, good. Sanya,
Yes, ma'am, it's working.
okay, good. Namrata,
Ma'am,
yeah.
I had a query that I have installed Cufflink through the source code, and it's opening Cuff Merge and Cuff Div is also opening, ma'am. So is there any problem?
Actually, why I'm telling you these steps, the thing is when you will run the pipeline, it won't run properly. So now like it is opening, but once you will run the pipeline, it won't, because you need to put in the bash rc file. Why we are putting this path in the bash rc? We are making this tool globalized. Okay, that's the reason, like we are putting the path in the bash rc.
Okay.
So for now, it will open like when you are opening the terminal from inside the tool. But when you will open from the raw data, it won't work there. You need to follow those steps.
Okay, ma'am.
So it's not about only about the cuffling. So for every source code, you need to make it globalized. Okay.
Yes, ma'am.
So yeah. Once it is done, let me know.
Do I need to paste the exact version in the code, or do I just need to type Python 2 or Python 3?
Only Python 2. We are not dealing with the Python 3 here. Just share your screen, Pushpit. Otherwise, it will take lot of time.
Ma'am, which one do I need to download of these?
No, 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 Pushpit. From the terminal. Just open the terminal. Just write sudo space, not here, below. Control Z, make it Control Z.
I did it. It's not.
Just press
Right.
Control Z. Is it working?
No, it's not.
Cancel this terminal. Just open the new one. Hmm. Yeah, just write here in the terminal sudo apt -get install Python 2. No, no, no. Don't give a space there. Yeah, enter. Why?
Now do I need to
Yeah,
just
then just open the tool, the cufflink one.
in the terminal only, right?
No, no, not here, Pushpit. Go inside the folder.
Okay.
Hmm.
Now I need to paste the.
Just open, yeah, go to tool. Just open this. Yeah, just open the cuff merge file. Yeah. So here you can see there is written Python only, right? So
Yeah.
just type here 2, save it. Yeah. Then open the terminal. Just write here cuff merge, cuff div.
Is that okay?
Yeah. So you can see the manual is open. So it's now working. Okay.
Okay.
Clear, everyone? Okay. So now Cufflinks is installed. So can you guys tell me what is this Cufflinks will do now? What is the role of the Cufflink here?
Ma'am, it will make the HISAT tool more accurate. Like the output will be better than not using Cufflinks.
Okay. So actually, Cufflink is a very important tool for studying the gene expression, alternative splicing, or maybe the gene regulation. Why? Because it is providing you valuable insight into the transcriptome. So basically, what it is doing, it is assembling what the fragmented RNA sequence reads into the continuous transcript and accounting for the alternate splicing and the gene fusion here. So can we say like whenever our gene is getting expressed, transcript will express, right?
Can you repeat the last statement?
Yeah. So ma'am, like basically in the cufflinks, what it is happening, it is assembling the fragmented RNA secrets into the continuous transcript and it is accounting for the alternate splice or the gene fusion here. So whenever our gene is expressing, so can we say the transcript will express? So here, whatever the result will get after the cufflinks, that would be the transcript file. So in those files, we'll get the count of each sample or the read, you can say. So we know like there are multiple reads, right? So they will map here and there. So due to that, we might get biasness in the data, right? So if there is an error or the biasness in our data, we need to remove that, right? Because manually we cannot remove the biasness from our data. So cufflinks will do the normalization in our data. So can anyone tell me what is this normalizing? Anyone?
And normalizing means we can compare all the data together. Like,
Okay. So actually, in the cufflinks, the normalization basically refers to the process of adjusting the expression value of transcript to account for difference in the sequence depth and the transcript length. So this is essential for accurately comparing the expression level between the different sample. So I already told you what happened whenever there are multiple reads, they will map here and there. So there might be get the error or the biasness. So there are two type of factors that are technical and biological. So these variation can impact the accuracy of the result. So in this technical variation, we will study about the sequence depth. So what happened in the sequence of the depth, the number of read sequence can affect the sensitivity of the cufflinks in detecting the low abundance transcript. So higher the sequencing depth generally leads to better detection. So for example, so this is my control sample and this is my treated. And if we are providing 30x depth to this control and 50x depth to this treated. So can anyone tell what it is explaining us?
That on an average, each nucleotide was sequenced at least 30 times.
Okay. So as we are having more reads in the treated sample, why? Because we are providing here 50x depth to this, right? More number of reads here. So can we say more number of genes is going to express in the treated sample, right? So it can give the error due to the different depth between the sample. So if the more number of reads are mapping on a reference genome, so it doesn't mean we can say the gene are expressing more from the treated sample. So that is why these depth are also leading to the biasness in our data. Clear this the depth part?
Yeah, ma'am.
Then
And should it
another.
not be same? Uh, uh, Depth should be same for both, right? If we are comparing them.
No, ma'am. If like by chance we are providing, it's up to you only whether like you want to treat the control sample with the same or different. It's up to you, ma'am. So next is the sequencing error rate. So error in the sequencing process can also give us the noise into the data. So basically, which will lead to the incorrect transcript assembly. Okay. So error in the process can lead to the noise. Next is the read mapping quality. So the quality of the read alignment to the reference genome can also influence what the accuracy of the transcript assembly and the quantification. Clear?
Can you repeat?
Mhm. The quality of reads alignment to the reference genome can influence the accuracy of transcript assembly and quantification. Okay. Now next is the biological variation. So in this, first thing is sample heterogeneity. So biological variation within a sample can make it difficult to accurately quantify the transcript expression level. Okay. Then next is the RNA degradation. So RNA degradation can also affect the representation of the transcript in the RNA-seq, and which will lead to the biased expression. So gene fusion can also create a challenge for the transcript assembly, as cufflinks may not be able to reconstruct the fused transcript. Okay? Should I write or like it is okay?
Ma'am, alternative splicing, can you repeat?
No, I haven't discussed till here. Is it fine till gene fusion?
Yes, ma'am.
Okay. So next is the alternatively splicing. The presence of this alternative splicing events can complicate the transcript assembly and the quantification, as cufflinks may not always correctly identify all the isoforms. So that is why these variations are leading to the biasness in our data. So to minimize this impact of variation, it is very important. So use the high quality RNA seq data, which will ensure that sequencing library preparation is done carefully and that sequence quality is high. And also, you can use the appropriate normalization method to account for difference in sequencing depth and the transcript length. Okay, clear? So now we have discussed what the factors, or you can say the variations, that will lead to the biasness, right? So these error and the biasness will be normalized by the cufflinks data, okay, or you can say the cufflinks tool. And we are getting the normalized data in the FPKM value. So what is this FPKM value? You might have seen in the NCBI also.
No, ma'am, no idea.
So it stands for fragment per kilobase million. So basically, it is a unit to measure the relative abundance of transcript in the RNA seq. So it accounts for both the number of reads mapping to the transcript and the transcript length, and providing a more accurate measure of the expression levels compared to the simple read count. So for each the transcript, the cufflinks, what it will do, it will calculate what the FPKM with the help of the formula. So formula is FPKM equal to into asterisk So this is the FPKM formula. Whereas the C is the number of reads mapped to the transcript. Then N is the total number of mapped reads in the sample. Then L is the length of transcript in kilobase. Okay, done. So these are the variations or the biasness which will be normalized with the help of this FPKM values. Okay. So mostly like the gene expression you will get in this value FPKM. Clear? Now we will see how to run this Cufflinks tool. Are you on the same page, guys?
Yes, ma'am.
Okay. So first you need to call the Cufflinks tool. So here hyphen O is the output name. T is the again the threads you can implement here. Then hyphen G. In the hyphen G, you have to provide what? The GTF file which you have downloaded from the UCSC. So why GTF here?
Ma'am, because it contains the annotation.
Yeah, so whenever like reads are mapping on the reference genome, we need to know what are those genes and the functions, that all the annotation part we need to know. So that is why we are putting here the GTF file. And this is the input file name which you got from the SAM tools. Okay, so for each and every file, you have to make the command and just run it in the raw data folder. Just create this file.
Ma'am, can you repeat?
Yeah, so you need to call the cufflink tool here. Then hyphen O is standing for the output file. So you can put any name to this output file. Then hyphen P is the thread. Then hyphen capital G is the GTF file here. So we are providing for the annotation part. Then this is the input file here, which you got from the SAM tools. Okay. So for each file, you need to create the command in the script and just run it.
That
Ma'am, for me, for the control 3 file, it's saying cannot open alignment file for reading for some reason.
Just share your screen. I will share the command also. Control 3. From where you got this control 3 file? We are not having any control 3.
Oh, okay. Yeah.
So we are having two file for control, right? Control 1, 2 treated, you can write there.
Yeah, I saw it.
Okay, no problem.
Done, ma'am.
yeah, done.
Okay.
Done, ma'am.
Okay. Now, so everyone is having this folder with the name Cufflinks.
Yes, ma'am.
Shall we start?
Yes.
So I will open this one folder here in the cufflink control one. So you can see this cufflinks will give you expression at gene level also, isoform, skipped, and the transcript file also. So here in this cufflink file, the transcript file is very important. Okay, so further to calculate the differential gene expression, we are going to use this transcript file. Okay, so if I open this gene, so in this gene FPKM file, it will provide you what the FPKM of the transcript per million reads. Okay, value for each gene. So it will include what the gene ID. So here you can see the ID, right? And the class code is nothing. It will just give you the gene ID. Some of the researcher it is giving the gene ID also in order to like not to get confused with the other genes. So that time they are giving. Then there is a nearest ref ID. You can see the locus also, right? And now you can see the FPKM, conflow, confi. So what is this now? So conflow, this word. So basically it stands for fragment per kilobase of transcript per million mapped reads confidence interval lower bound. So it represents the lower limit of the range within which the true FPKM value for a particular transcript is expected to fall with a certain degree of confidence. So this is
Ma'am, can you
just
repeat what is the full form?
so this stand for fragment per kilo. This confidence interval high bound, it representing the upper limit of the range within which the true FPKM value for a particular transcript is expected to fall within a certain degree of confidence. Okay, so it is just the lower limit and it is just the higher limit. Okay, within this range, the transcript value, or you can say the transcript FPKM value, should fall. Clear?
Yes, ma'am.
Again, these isoforms also will contain the locus, the IDs, and the length. Again, this is the FPKM value, this is the lower limit, and this upper limit. But these are not the important files here. So this, what is a skipped GTF? Can anyone tell now? What is a skipped GTF here?
Ma'am, I think like these are the location where there was no alignment.
Yeah. So identify the region of transcript that were not covered by reads. Okay. Now if I open this, the transcript GTF file, so this is the important file. This file we are going to use in the cuff merge. So in this transcript.gtf file, it will contain all the assembled transcript structure in the GTF format. Can you see? This is the GTF format that you have also seen in the UCSC, which will include the chromosome location. This is the cufflink tool name. This is just a feature. This is the start and the end coordinate. This is the flag score, which is nothing, just a statistical score here. Then next, it will give you the strand information, then the frame information, These are journal attribute, or you can say the extra information they are giving you here. The gene ID, transcript ID, and you can see here the FPKM value it is calculated. And this is the again the lower limit and the higher limit, and this is the COV. So COV is again COV in the frac. This word is it refers to the coverage of the particular region or the feature in the reference sequence. Okay. Reference genome. So it indicates the average number of a time a nucleotide is that region has been sequenced. So that is what the curve. So higher COV suggests the higher sequencing depth and the greater confidence in the observed expression level. Whereas this frac refers to the fractional abundance of transcript. So that means a higher frac value will indicate a greater greater proportion of the total RNA in the sample, which is composed of the transcript. So clear? So it's all about the cufflinks.
Yes, ma'am.
So we will end this today's session and we'll meet on Monday. Till then, you can practice and study like regarding this.
Yeah, ma'am.
Chalo, see you guys. Bye bye. Take care.
Thank you, ma'am.